Welcome to our second video of Unit 2.7, okay, of the molecular biology topic. In this unit, we're looking at transcription. Well, in the whole unit, we've been looking at replication, transcription, and translation. So we've already looked at DNA replication in the previous video. Now we're looking at the processes of transcription and translation as we move from our DNA molecule through to our chain of amino acids, our polypeptide chain, which will form our protein. Okay, so transcription and translation. And when you look at the words, think of what these words mean before you even know any of the biology. Transcription, transcribe, to copy, okay, and translation, to translate. From one thing to another, to translate. Okay, so think of what those words mean to you. Alrighty, so in this infographic here, we can see our initial DNA molecule. Okay, now to the right there, it's saying that with replication, what then forms are two new DNA molecules. And remembering that's semi-conservative, so you've got one strand of the parent DNA molecule in each of those new DNA molecules. Transcription and translation is different. You actually end up with a completely different molecule. We start with DNA, we end up with a polypeptide, which then forms the protein. Okay, so we've got these two stages of getting from a DNA molecule or a section of DNA, which we'll call a gene, to our polypeptide chain and then our protein. And these two processes are our transcription and translation. So we know that our genetic information is encoded in DNA, okay, and that's been the biological theory for over 50 years now. And remembering, you may go into careers that prove these things wrong, or develop these theories further. This is an ever-changing science. Okay, now that our DNA is transcribed into molecules of RNA, and in our previous unit we looked at the differences between our DNA and our RNA. The reason that we have to transcribe to RNA is because DNA is actually too complex a molecule to fit through the nuclear membrane, whereas our RNA, being a single chain, is a simpler molecule and will fit through the membrane. Remembering DNA is a very, very long molecule. RNA is a copy of a gene, so only a section of that DNA. So it's a shorter molecule. It can get out the nuclear membrane to the ribosome, where our polypeptide formation occurs. So once we've got that RNA, which is a copy of the information in the gene in the DNA, that passes out of the nuclear membrane, heads to the ribosome, and it is at the ribosome that translation occurs, where that RNA is read and our polypeptide chain of amino acids is then formed. And you can see all that in the picture there. Okay, so let's think about this. We start off with our DNA. That becomes transcribed to what we call mRNA, so our messenger RNA. Now, if you look at that RNA molecule there, you can see uracil on it. And remember, in 2.6, we looked at the differences between DNA and RNA, and we said that DNA had the bases ATCG, whereas RNA had AUCG. Okay, so remember, on RNA, thymine is copied as uracil. Okay, so that's mRNA in the chain like that. We're going to come across a different type of RNA as well, which is our transfer RNA. And that's involved in the translation process. And it's the transfer RNA that codes for each amino acid. So we're going to look into that further as we go on. But this picture shows how that transcription is the movement from the DNA to the messenger RNA, and then translation occurs between the messenger RNA to a protein, with transfer RNA involved in that process. And then Our proteins can be catalytic, so like our enzymes, they can be transport, immunological, contractile, regulatory, or structural. There's so many different uses for proteins, and this is why proteins are coded by those amino acids that can be arranged in all different arrangements on the chain, and then forming so many different types of protein. Okay, now reverse transcription can occur, and we'll look at that at a later date. Alrighty, so types of RNA. We spoke about mRNA, so this is messenger RNA. This carries the genetic message to the copy of the gene from the DNA out to the ribosome, where that message can then be translated into a particular protein. We've got that transfer RNA. Those molecules carry the amino acids to the ribosome, and then that's where they construct the protein. We can also get ribosomal RNA, and together with proteins, these form the structure of the ribosome in the cytosol. Okay, so looking at transcription. So sections of the DNA that code for particular proteins called genes. We've looked at that. Okay, DNA is copied within the nucleus. We've looked at that. Genes contain specific base sequences in sets of 3. We call them triplets. Each triplet codes for a specific amino acid. So when we get to translation, we're looking at the order of our bases in 3s. Okay, so we look at the bases in triplets. And this then determines where transcription begins and ends, because we have start and stop triplets or codons. Okay. As DNA doesn't leave the nucleus, the code for a gene needs to be copied onto the mRNA. So we've said that DNA stays in the nucleus, mRNA, messenger RNA, carries that gene message out of the nucleus. This process, that's called transcription. Okay, so the copying of the gene from the DNA to the mRNA is the process of transcription. Okay, the mRNA then carries that code out of the nucleus to the cytoplasm, where it's heading to the ribosome, ready to make our polypeptide. Here you can see an image of transcription, and we've got that enzyme here, RNA polymerase. Okay, and this is what's attaching those 3 nucleotides to the RNA chain, okay, and making that RNA chain, so bonding those molecules. Let's have a look at mRNA in RNA polymerase in action. There it goes. Off it whizzes, and you see that long chain there of the gene that's being copied into that mRNA. So that yellow is the mRNA. So cool. So that DNA is being unzipped, and one chain of it is being read, so that the mRNA can be formed. And those yellow 3 nucleotides floating around there, so that it's got access to the ones that it needs to do that base pairing. Okay, let's have a bit of a look at transcription a little bit deeper. So DNA is unzipped by the mRNA. Okay, so the mRNA here has got quite a few jobs. The mRNA is breaking those bonds and unzipping. So in DNA, helicase unzipped. In the DNA in DNA replication, we had helicase unzipping. In transcription, we've got RNA polymerase doing the unzipping. Okay, now it's going to break those hydrogen bonds so that it unzips that DNA. And that mRNA and the RNA polymerase knows the start and stop triplet to know which part of the gene it's going to be unzipping. It's not going along the entire length of the DNA, remember, whereas helicase unzip the whole lot. RNA polymerase is only unzipping the section of the DNA that is the gene that it needs to copy. Now, we've got our 3 RNA nucleotides. They're floating around in the nucleus. Okay, and they're going to come along. They pair up with the strand of DNA that's being copied. So that section of gene, they come along, base pairing. Remember, we're now making a molecule of mRNA. So uracil is going to pair with adenine. Okay. The RNA polymerase, as those 3 nucleotides pair up, is then catalyzing the chain to make the mRNA polymer. That then forms that single strand of mRNA, some messenger RNA. The mRNA then separates from the DNA, which then zips back up into its double helix. Okay. The mRNA is then going to leave the nucleus by moving through a pore in the nuclear envelope out into the cytoplasm. Where does it head when it's in the cytoplasm? It's heading to a ribosome. And on the ribosome, that's when translation will occur. So we've got another image there showing our transcription. Okay, now the DNA strand with the same base sequence as the mRNA, we call that the sense strand. Okay, so the chain that has the same sequence as the mRNA is the sense strand. The other strand that acts as the template and has the complementary base sequences, so the one that's the template, the one that the mRNA is copied from, we call the antisense strand. So you see in our image here, we've got that RNA polymerase, and it's got these dual roles. It unzips, and then it joins the 3 nucleotides together to make the RNA strand. Okay, so we've copied the gene. Our mRNA is a copy of the gene. It's left the nucleus via a nuclear pore into the cytoplasm. It's now heading for the ribosome. Before we get to the actual process of translation, little quick bit of amino acid revision. Okay, our structure of our amino acids, we know each amino acid is different because they have different R groups. This was our table of our 20 most common amino acids that we looked at. You will need those 3-letter codes will be quite important in protein synthesis because we're not writing those full names out. Okay, 2 amino acids joined together form a polypeptide bond between them. A chain of amino acids then are called a polypeptide. It is not a protein until it's those tertiary and quaternary changes have occurred to fold it into the protein. Alrighty. Translation is the process where the coded information on the mRNA strand is used to construct a polypeptide chain, which will form a functioning protein. This happens in the cytoplasm at the ribosomes. Alrighty, let's get this show on the road. Earlier on, I talked about triplets, so 3 bases together. Okay, now this is what I'm talking about. Have a look here at our mRNA strand. 3 bases together there, and in this example, we've got guanine, cytosine, and uracil. Those 3 bases in these triplets we call codons. And those 3 bases code for specific amino acids. Okay. So each triplet of bases on the mRNA is called a codon, and each codon codes for 1 amino acid. Alrighty, so here in this image, this is our mRNA, our messenger RNA. So that's that section of gene that has been transcribed from the DNA out in the cytoplasm at the ribosome, waiting to be read. Along comes now tRNA, so transfer RNA. On the end of the transfer RNA are 3 bases. And what they do is they will match up with their corresponding bases on the mRNA. On the other end of the tRNA is the matching amino acid. So for example, the codon UUU matches, so uracil, 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 matches to the amino acid lysine, okay? CUA, aspartame. I don't know all my amino acids off the top of my head just like that. I'm trying. Okay, but basically, the codons on the base of the tRNA code for a specific amino acid. There'll be, once that amino acid's taken off the end of the tRNA goes off into the cytoplasm, finds another amino acid that matches its bases. Okay, so they come along, match up, drop their amino acid, and buzz off and find another amino acid out in the cytoplasm. Basically, that's maybe not quite biologically exact, but that's the basic principle. Okay, so ribosomes bind to the mRNA. So the mRNA is in place. The specific tRNA, so transfer RNA, with complementary anticodons. So we've got our codons on our mRNA and our anticodons on our tRNA are drawn into position by the ribosome. Each tRNA will carry a specific amino acid with a matching triplet pair. The anticodon of the tRNA binds to the complementary codon of the mRNA with very weak hydrogen bonds. So it's those hydrogen bonds between bases, those base pairs. When two tRNA are in place, a peptide bond will be formed between those 2 amino acids. So we've got these 2 in place. Our peptide bond is forming between them. When we've got 2 attached, we've got a dipeptide. Then when we get more attached, we're forming that polypeptide. The first tRNA will then detach from its amino acid. So this one's going to detach. The ribosome will then move along the mRNA to the next codon where the process is repeated. So this tRNA will stay there until this one's in place. They've got their peptide bond, and then this one will stay there and that one will buzz off. The final codon is called a stop signal, okay, which tells the ribosome to detach from the mRNA. The peptide then floats free into the cytoplasm, ready to start its formation into the protein. Okay, let's watch this in action. So we've got our start and stop codons. Here comes our ribosome. So let's watch it again. Along comes our ribosome. We've got our start codon, okay, and here's our first amino acid. Coming along, pairing our stop codon, and off goes our protein. Our polypeptide. Okay, so here we've got a table. Now, it might look a bit daunting and confusing to start off with, but don't be confused, okay? Imagine we've got our mRNA, so that first copy that's come from our gene on the DNA. So we're going to be looking at these codons, these triplets of bases on our mRNA. So if we look here, the first of the triplet, the first base of the triplet, for example, if it was U, and then our third letter was U, here's the amino acid it codes to. We've got a triplet, which is, I'm just going to pull 3 letters out the air here, ACG. Let's have a look. So our first one's A, C, so A, C, C, G. There we go. There's our amino acid. All right, 3 others. C, G, C, 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 G. So I've gone across C, down G, C, G, C. There we go. Codes for that amino acid. So that's how we use that table. We'll do some practice in class. Don't panic. Alrighty. Let's have a watch of this little video.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acid, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So that video gave a brief overview and a good infographic around how the polypeptide chain is formed. There are some new terms in there that are good to know, but not necessarily linked to your course, okay? All right, so one gene, one polypeptide. Generally, it's agreed that only one gene codes for our polypeptide, but not necessarily one protein, okay? Remembering that sometimes several polypeptides join together to form our protein. And those different polypeptides would come from different genes. Some DNA sequences act as regulators for other genes and are not transcribed and translated themselves. So not all of the DNA will be used through transcription and translation. Some mRNA strands form a single gene. Forming a single gene can be modified differently by having different segments reassembled in a different order, resulting in different polypeptides being formed. and some genes will be expressed differently in different tissues or at different stages of their development. Okay, so that's everything on this unit so far. We'll cover the rest of it in class. See you in our next video.